Hello dear students. Today the theme of our lecture is called DNA repair, and we will talk about protein biosynthesis too. Here is two themes to our learning. So, our questions on lecture today. The first question, it is a DNA repair. And we will be talk about two types of DNA repair, which called proofreading and mismatch repair. After that, we will talk about DNA damage repair pathway, such as direct reversal, excision repair, and double-stranded break repair. And after that, we will talk about the meaning of the DNA repair and proofreading in the course of human diseases. And after that, we will talk about the process of protein biosynthesis, about the stage of protein biosynthesis. So let's start. As you know, in our organism, mutations are actually happening all the time. However, these mutations don't cause cancer or another pathology. Why? That's because they are usually detected and fixed by DNA proofreading and repair mechanism. Or if the damage cannot be fixed, the cell will undergo programmed cell death, which called apoptosis, and avoid passing on the incorrect DNA. Mutations happen and get passed on the daughter cells only when this mechanism fails. Today we'll take a closer look at the mechanism used by cells to correct replication errors and fix DNA damage, including proofreading, for example, the first proofreading. Proofreading, which corrects errors during DNA replication. Also, we will talk about mismatch repair, which fixed mispaired bases right after DNA replication, and about repair pathway of the DNA damage, which detected and correct damage throughout the cell cycle. I remind you that the DNA polymerases are the enzymes that build DNA in cells. During DNA replication or copying DNA, most DNA polymerases can check their work with each base that they add. This process is called proofreading. If the polymerase detects that a wrong or incorrectly paired nucleotide has been added, it will remove and replace the nucleotide right away before continuing with DNA synthesis. Mismatch repair. Many errors are corrected by proofreading, but a few slip through. Mismatch repair happens right after new DNA has been made, and its job is to remove and replace mispaired bases, ones that were not fixed during proofreading. First, protein complex recognizes and binds to the mispaired base. A second complex cuts the DNA near the mismatch, and more enzymes chop out the incorrect nucleotide and the surrounding patch of the DNA. A DNA polymerase then replaces the missing section with correct nucleotides, and an enzyme called a DNA ligase seals the gap. DNA damage repair mechanisms. Bad things can happen to DNA at almost any point in a cell's lifetime, not just during replication. In fact, your DNA is getting damaged all the time by outside factors like UV light, chemicals, and X-rays, not to mention spontaneous chemical reactions that happen even without environmental insults. Fortunately, your cells have repair mechanism to detect and correct many types of DNA damage. Repair processes that help fix damaged DNA include direct reversal. Some DNA-damaging chemical reaction can be directly undone by enzymes in the cell. Excision repair. Damage to one or a few bases of DNA is often fixed by removal or excision, in other words, and replacement of the damaged region. In base excision repair, just the damaged base is removed. In nucleotide excision repair, as in mismatch repair, we saw above, a patch of nucleotides is removed. And double-stranded break repair. Two main pathways, non-homologous and joining and homologous recombination, are used to repair double-stranded breaks in DNA. That is when an entire chromosome splits into two pieces. Reversal of damage. In some cases, a cell can fix DNA damage simply by reversing the chemical reaction that caused it. To understand this, we need to realize that DNA damage often just involves an extra group of atoms getting attached to DNA through a chemical reaction. For example, guanine can undergo a reaction that attaches a metal group to an oxygen atom in the base. The methyl-bearing guanine, if not fixed, will pair with the thymine rather than cytosine during DNA replication. Likely, humans and many other organisms have an enzyme that can remove the methyl group, reversing the reaction and returning the base to normal. Base excision repair. Base excision repair is a mechanism used to detect and remove certain types of damaged bases. A group of enzymes called glycosylases play a key role in base excision repair. Each glycosylase detects and removes a specific kind of damaged base. For example, a chemical reaction called deamination can convert a cytosine base into uracil, a base typically found only in RNA, as you know. During DNA replication, uracil will pair with the adenine rather than guanine, as it would if the base was still cytosine. So an uncorrected cytosine to uracil change can lead to a mutation. To prevent such mutation, a glycosylase from the base excision repair pathway detects and removes deaminated cytosine. Once the base has been removed, the empty piece of the DNA backbone is also removed and the gap is filled and sealed by other enzymes. Nucleotide excision repair. Nucleotide excision repair is another pathway used to remove and replace damaged bases. Nucleotide excision repair detects and corrects types of damage that distort the DNA double helix. For instance, this pathway detects bases that have been modified with bulky chemical groups, like the ones that get attached to your DNA when it's exposed to chemicals in cigarette smoke. Nucleotide excision repair is also used to fix some types of damage caused by UV radiation, for instance, when you get a sunburn. UV radiation can make cytosine and thymine bases react with neighboring bases that are also cis or ts, forming bonds that distort the double helix and cause errors in DNA replication. The most common type of linkage, a thymine dimer, consists of two thymine bases that react with each other and become chemically linked. In nucleotide excision repair, the damaged nucleotides are removed along with the surrounding patch of DNA. In this process, a helicase or DNA opening enzyme cranks open the DNA to form a bubble, and DNA coding enzymes chop out the damaged part of the bubble. A DNA polymerase replaces the missing DNA, and the DNA ligase seals the gap into the backbone of the strand. And our last type of the mechanism, it is a double-stranded break repair. Some types of environmental factors, such as high energy radiation, can cause double-stranded breaks in DNA, splitting a chromosome in two. This is the kind of DNA damage linked with superhero origin stories in comic books and with disasters like Chernobyl in real life. Double-stranded breaks are dangerous because large segments of chromosomes and the hundreds of genes they contain might be lost if the break is not repaired. Two pathways involved in the repair of double-stranded DNA breaks are the non-homologous end joining and homologous recombination pathway. In non-homologous end joining, the two broken ends of the chromosome are simply glued back together. This repair mechanism is missing and typically involves the loss or sometimes addition of a few nucleotides at the cut site. So non-homologous end joining tends to produce a mutation, but this is better than the alternative. I mean, loss of an entire chromosome arm. In homologous recombination, information from the homologous chromosome that matches the damaged one, or from a sister chromatid if the DNA has been copied, is used to repair the break. In this process, the two homologous chromosomes come together, and the undamaged region of the homolog or chromatid is used as a template to replace the damaged region of the broken chromosome. Homologous recombination is cleaner than non-homologous end joining and does not usually cause mutation. And now let's talk about the role of the DNA repair in the coding of different human diseases. Evidence for the importance of proofreading and repair mechanism comes from human genetic disorders. In many cases, mutations in genes that encode proofreading and repair proteins are associated with hereditary cancers that run in families. For example, hereditary non-polyposis colorectal cancer, also called Lynch syndrome, is caused by mutations in genes and encoding certain mismatch repair proteins. And the next example, it is xeroderma pigmentosum. People with xeroderma pigmentosum are extremely sensitive to UV light. This condition is caused by mutation affecting the nucleotide excision repair pathway. When this pathway does not work, thymine dimers and other forms of UV damage can be repaired. People with xeroderma pigmentosum develop severe sunburns from just a few minutes in the sun, and about half will get skin cancer by the age of 10 unless they avoid the sun. So, our theme about the DNA repair is over, and our second part of our lecture will be about protein synthesis. So, protein synthesis is a process of creating protein molecules. And as you know, the information about structure of the protein are allocated in DNA, and DNA located in the nucleus. So the first stage of the of the protein synthesis is called nuclear phase, which include two proteins, transcription and processing. Transcription consists of three main parts. It is initiation, elongation, and termination. And process of processing consists of three parts. It's called modification of the ending. It means modification of the ending of mRNA. Excision of the introns. I remind you that the introns, it is a part of the genes without the informations. And the last stage, it is the splicing of the exons. Exons, I remind you, that is a part of the genes with genetic information about the structure of the proteins. And after nuclear phase of protein synthesis, then starts the plasmic phase of the protein synthesis, which happened in the cytoplasm. And the plasmic phase also include two big stages, which called translation and post-translational processes. And how you can see, the translation includes stage 2, like transcription, and this process has the same name. So, initiation, elongation, and termination. And on the slide you can see some example of post-translational processes such as proteolysis, different chemical modification or chemical reactions such as methylation, ubiquitination, phosphorylation, acetylation, glycosylation, and folding. And now let's talk about these processes in the detail. So, the first process of the nuclear phase of protein biosynthesis is called transcription. What means transcription? You know that the transcription is a process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or mRNA. The enzyme, the main event of the initiation of transcription is when the enzyme RNA polymerase binds to the exposed template strand and reads from the gene into 3 to 5 direction. And the next stage is called elongation. Simultaneously, the RNA polymerase synthesis a single strand of pre-mRNA in the 5 to 3 direction, by catalyzing the formation of phosphodiester bonds between activated nucleotides free in the nucleus that are capable of complementary base pairing with the template strand. The pre-mRNA molecular synthesis is complementary to the template DNA strand and shares the same nucleotide sequences as the coding DNA strand. However, there is one crucial difference in the nucleotide composition of DNA and mRNA molecules. DNA is composed of the base guanine, cytosine, adenine, and thymine, and RNA is also composed of 4 bases but guanine, cytosine, adenine, and uracil. In RNA molecules, the DNA base thymine is replaced by uracil, which is able to base pair with adenine. Therefore, in the pre-mRNA molecule, all complementary bases, which would be thymine in the coding DNA strand, are replaced by uracil. And the last stage of the transcription is called termination. When RNA polymerases reaches a specific DNA sequence which terminates transcription, RNA polymerase detaches and pre-mRNA synthesis is complete. And after that, processing is occurred. Once transcription is complete, the pre-mRNA molecule undergoes post-transcriptional modification to produce a mature mRNA molecule. There are 3 key steps within post-transcriptional modification. Addition of 5 cap to the 5 ends of the pre-mRNA molecule. Addition of a free poly A tail is added to the free end pre-mRNA molecule. And the last, removal of introns via RNA splicing. The 5 cap is added to the 5 ends of the pre-mRNA molecule and is composed of a guanine nucleotide modified through methylation. The purpose of the 5 cap is to prevent breakdown of the mature mRNA molecules before translation. The cap also aids binding of the ribosome to the mRNA to start translation and enables mRNA to be differentiated from other rRNAs in the cell. In contrast, the free poly A tail is added to the free end of the mRNA molecule and is composed of 100 to 200 adenine bases, so it is called poly A or polyadenine. This distinct mRNA modification enables the cell to detect that the full mRNA message is intact if both the 5 cap and free tail are present. This modified pre-mRNA molecule then undergoes the process of RNA splicing. Genes are composed of a series of introns and exons, as we know. Introns, I repeat that the introns are nucleotide sequences which do not encode a protein, while exons are nucleotide sequences that directly encode a protein. Introns and exons are present in both the underlying DNA sequences and the pre-mRNA molecule. Therefore, in order to produce a mature mRNA molecule encoding a protein, splicing must occur. During splicing, the intervening introns are removed from the pre-mRNA molecule by a multiprotein complex known as a spliceosome, composed of over 150 proteins and RNA. This mature mRNA molecule is then exported into the cytoplasm through nuclear pores in the envelope of the nucleus. And after that, there are cytoplasmic stage of the protein biosynthesis, and the first stage of the plasmic stage is called translation. In translation, the sequences of the mRNA is decoded to specifically the amino acid sequences of a polypeptide. And the genetic code is the set of the rules used by living cells to translate information encoded within genetic material, DNA or mRNA sequences of nucleotide triplets or codons, into protein. So, what are the main properties of the genetic code? Universal. What does it mean? Genetic code is universal. All non-living organisms use the same genetic code. This shows that all organisms share a common evolutionary history. The second property of genetic code is specificity. Each codon codes for just one amino acid. The next property is degeneracy. Most amino acids are encoded by more than one codon. Non-overlapping. Adjacent codons do not overlap. It means a non-overlapping code means that the same letter is not used for two different codons. In other words, no single base can take part in the formation of more than one codon. And the last properties of the genetic code that it reads without punctuations. What does it mean? There is no signal to indicate the end of one codon and the beginning of the next. The genetic code is commaless or comma-free in other words. During translation, ribosomes synthesize polypeptide chains from mRNA template molecules. We said it. In eukaryotes, translation occurs in the cytoplasm of the cell, where the ribosomes are located either free-floating or attached to the endoplasmic reticulum. And we said it's why the translation is the stage of the cytoplasmic stage of the protein biosynthesis. The mRNA nucleotide sequences is read in triplets. 3 adjacent nucleotides in the mRNA molecule correspond to a single codon. Each tRNA has an exposed sequences of 3 nucleotides known as anticodons, which are complementary in sequences to a specific codon that may be present in mRNA. For example, the first codon encountered is the start codon composed of nucleotides AUG. The correct tRNA with the anticodon complementary through nucleotide sequences URC binds to the mRNA using the ribosome. This tRNA delivers the correct amino acid corresponding to the mRNA codon. In the case of the start codon, this is the amino acid methionine. The next codon, adjacent to the start codon, is bounded by the correct tRNA with complementary anticodon, delivering the next amino acid to ribosome. The ribosome then uses its peptidyltransferase enzymatic activity to catalyze the formation of the covalent peptide bond between the two adjacent amino acids. The ribosome then moves along the mRNA molecule to the third codon. The ribosome then releases the first tRNA molecule, as only two tRNA molecules can be brought together by a single ribosome at one time. The next complementary tRNA with the correct anticodon complementary to the third codon is selected, delivering the next amino acid to the ribosome, which is covalently joined to the growing polypeptide chain. Termination of the growing polypeptide chain occurs when the ribosome encounters a stop codon, such as UAA, UAG, or UJR in the mRNA molecule. When this occurs, no tRNA can recognize it, and a release factor induces the release of the complete polypeptide chain from the ribosome. Once synthesis of the polypeptide chain is complete, the polypeptide chain folds to adopt a specific structure which enables the protein to carry out its function. The basic form of protein structure is known as the primary structure, which is simply the polypeptide chain, a sequence of covalently bonded amino acids. The primary structure of a protein is encoded by a gene, therefore any changes to the sequences of the gene can alter the primary structure of the protein and all subsequent levels of protein structure, ultimately changing the overall structure and function. The primary structure of a protein or polypeptide chain can then fold or coil to form the secondary structure of the protein. The most common types of secondary structure are known as an alpha helix or beta sheet. These are small structures produced by hydrogen bonds forming within the polypeptide chain. This secondary structure then folds to produce the tertiary structure of the protein. The tertiary structure is the protein's overall 3D structure, which is made of different secondary structures folding together. In the tertiary structure, key protein features, the active sites are folded and formed, enabling the protein to function. Finally, some proteins may adopt a complex quaternary structure. Most proteins are made of a single polypeptide chain. However, some proteins are composed of multiple polypeptide chains, known as subunits, which fold and interact to form the quaternary structure. Hence, the overall protein is a multi-subunit complex composed of multiple folded polypeptide chain subunits hemoglobin. When protein folding into the mature functional 3D state is complete, it is not necessarily the end of the protein maturation pathway. A folded protein can still undergo further processing through post-translational modification. There are over 200 known types of post-translational modification. This modification can alter protein activity, the ability of the protein to interact with other proteins, and where the protein is found within the cell, in the nucleus or cytoplasm. Through post-translational modification, the diversity of proteins encoded by the genome is expanded by 2 to 3 orders of magnitude. There are 4 key classes of post-translation modification. Proteolysis or cleavage of proteins is an irreversible post-translational modification carried out by enzymes known as proteases. The proteases are often highly specific and cause hydrolysis of a limited number of peptide bonds within the target protein. The resulting shortened protein has an altered polypeptide chain with different amino acids at the start and the end of the chain. This post-translational modification often alters the protein's function. The protein can be inactivated or activated by the cleavage and can display new biological activities. Following translation, small chemical groups can be added onto amino acids within the natural protein structure. Examples of proteins which add chemical groups to the target protein include methylation, acetylation, and phosphorylation. Methylation is the reversible addition of methyl group onto an amino acid catalyzed by methyltransferase enzymes. One example of a protein which is commonly methylated is a histone. Acetylation is the reversible covalent addition of an acetyl group onto an amino acid by the enzyme acetyltransferase. The acetyl group is removed from a donor molecule known as acetylcoenzyme A and transferred onto the target protein. Histones undergo acetylation on their lysine. Charge interaction between lysine residue by enzymes known as histone acetyltransferase. The effect of acetylation is to weaken the charge interaction between the histones and DNA, thereby making more genes in the DNA accessible to transcription. And the last type of the post-translational process by addition of chemical groups is phosphorylation. Phosphorylation is a reversible covalent addition of a phosphate group to specific amino acid such as a rule serine, threonine, and tyrosine within the protein. The phosphate group is removed from the donor molecular ATP by protein kinase and transferred onto the hydroxyl group of the target amino acid. This produced adenosine diphosphate as a B product. This process can be reversed and the phosphate group removed by the enzyme protein phosphatase. Phosphorylation can create a binding site of the phosphorylated proteins, which enables it to interact with other proteins and generate large multi-protein complexes. Alternatively, phosphorylation can change the level of protein activity by altering the ability of the protein to bind its substrate. Post-translational modification can incorporate more complex large molecules into the folded protein structure. One common example of this is glycosylation, the addition of polysaccharide molecule. which is widely considered to be most common post-translation modification. In glycosylation, a phosphoride molecule known as a glycan is covalently added to the target protein by glycosyltransferases enzymes and modified by glycosidases in the endoplasmic reticulum and Golgi apparatus. Glycosylation can have a critical role in determining the final folded feeder structure of the target protein. And the last type of post-translational processes is called ubiquitination. Ubiquitination is an enzymatic process in which ubiquitin protein is attached to a substrate protein. And now let's repeat all our information and look some funny.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Today our lecture is over. I will hope to see you in the next week and have a good time. Thank you. Goodbye.